Hello members, to online once again, select Alan to get his name. We are back. We previously looked at the agents of pollination, and we are specifically going to look at now the process of fertilization in a flowering plant. This is what we are specifically going to look at. But before we go into this, we very well know that the fertilization involves fusion of the male reproductive gametes with the female reproductive gametes. The male reproductive gametes in plants, the male gametes are the pollen grains, and the female gametes are the egg cells inside the ovules. So before we go in further, let's first look specifically how do the ovules look like? How do the pollen grains look like? So let's look at the structure of a pollen grain first, because we are going to need this information as we describe this process. A pollen grain has got a smooth inner membrane, very smooth and thin, and this inner membrane is surrounded by a sculptured rough membrane outside we call the exine. It is rough, that's why I'm using this appearance, and this outer membrane is known as the exine. It is rough and tough. Two, the inner membrane is known as the entine. The entine. Inside this inner membrane is surrounding the entine is surrounding what we call the pollen cell cytoplasm, which is surrounding two cells. It is surrounding the polar tube nucleus and the generative cell, which is larger. So this is the pollen tube nucleus, and this is the generative cell or the generative nucleus. Actually, inside the generative cell, there is a nucleus. So this one becomes the generative cell nucleus, and this is the generative cell. This one is the generative cell nucleus. So this is the pollen tube nucleus, because this is the pollen tube cytoplasm. If we can remember very well, for those who covered this information. The structure of an ovule is made up of outer membranes we call integuments. The outer membrane, it is a double layered membrane, and this is called an integument with a small opening down here, and then a short stalk that attaches it to the wall of the ovary. Inside this, there is a very large nucleus. This is the nucleus of this ovule, which is surrounding a certain small cell-like structure we call, surrounding a small membrane we call the embryo sac. This is a small membrane surrounded by the nucleus of the ovule. This small opening is known as the micropyle. We said there is space above the nucleus, and it is the chalaza, the cell chalaza of the ovule. I think we are following. We said this double-layered membrane is known as the integument. It is the integument. Inside the embryo sac, there is a number of cells. The cell just close, the large cell just close to the micropyle is known as the egg cell. It has its own nucleus. And it also has other cells besides it, and they are two in number. And these cells are known as synagogues. These two cells. So this is a synagogue, this one is also a synagogue. This cell that is close to the opening is what we call the egg cell. It has its own nucleus. In the middle of the embryo sac, there are two nuclei. There is a pair of nuclei we call the polar nuclei. And at the top of the embryo sac, we have three cells. And these three cells are known as antipodal cells. So these are some of the things that make up the ovule. We have the integuments, we have the chalaza, the embryo sac, the antipodal cells, the polar nuclei, the egg cells, the synagogues, which are two in number, and the integuments with a micropyle at the opening. So this is the structure of an ovule. Before we go into the other process, it was very crucial for us to know this one. So, and below is the process of fertilization. We are going to look at what happens after pollination. The pollen grains have been transferred safely to a mature stigma of a flower of the same species. What happens? What happens until when the fruits are formed? It is what we are talking about. So, immediately, when a mature and ripe pollen grain lands onto a mature and ripe stigma, once it lands on a stigma that is ripe and mature, what it does, it absorbs water and mineral salts. The pollen grain will absorb water and mineral salts, and this is from the what? From the stigma. That is what it does once it lands on the stigma. The moment it absorbs this water and mineral salts from the stigma, it starts to germinate. It germinates. As it germinates, it forms what we call a pollen tube. A pollen tube. This pollen tube is led by the pollen tube nucleus, and it starts to grow downwards the stigma, downwards towards the ovary. If this is the stigma, once a pollen grain lands on it like this, it starts to absorb water and mineral salts from it. Once it absorbs water and mineral salts, it germinates to form what we call a pollen tube that grows down in the style towards the ovary. This pollen tube is led by a pollen tube nucleus. So at the bottom or at the tip of the pollen tube that is growing down in towards the ovary, there is a pollen tube nucleus that we saw in the pollen grain. And behind the pollen tube nucleus, there is this generative cell nucleus. As the pollen tube is growing down towards the ovary, this generative cell nucleus divides by mitosis. Mitosis, I mean the cell divides into two equal halves. After that division, it forms two meronuclei, which continues to grow, to follow the pollen tube nucleus as it enters the ovary. Once the pollen tubes are in the ovules through the micropyle, let me use this one as an illustration. Once this pollen tube has grown into this ovule through this micropyle, the tip of this pollen grain will rupture. Rupturing means it will break. The tip will break. And once this tip breaks, the pollen tube nucleus will disappear. When the pollen tube nucleus disappears, remember, there are two meronuclei following the pollen tube nucleus that were made as a result of division of this generative cell nucleus. One of the meronuclei will fuse with the egg cell to form a zygote. And then the second meronucleus that is remaining will fuse with the two polar nuclei to form what we call a triploid endosperm, a triploid primary endosperm, which later develops into a secondary endosperm that stores food for the seed. As development goes on, the zygote undergoes repeated divisions until when it becomes an embryo, which is made up of the primule and the radicle, which during the process of germination, it becomes a seedling. As simple as that. We've said once a mature and ripe pollen grain lands onto a ripe and mature stigma, it absorbs water and mineral salts. Once it absorbs the water and the mineral salts, we've said the pollen grain will start to germinate. The pollen grain will germinate into a pollen tube. The pollen tube will start to grow down in the style towards the ovary, led by the pollen tube nucleus, as the generative cell nucleus is lying behind it. As the pollen tube grows down towards the ovary, the generative nucleus divides by mitosis to form two meronuclei, which once inside the pollen, inside the ovule through the micropyle, the pollen tube tip ruptures such that the pollen tube nucleus disintegrates or disappears. As the pollen tube nucleus disappears, one of the meronuclei that were formed after the division of this generative cell nucleus will fuse with the egg cell to form a zygote, a diploid zygote, that later develops into an embryo. And then the second meronucleus will fuse with the polar nuclei to form a triploid endosperm, which becomes the storage site for the food in the seed after fertilization, after the seed has been fully formed. And simply that is the process of fertilization in a flowering plant. It's what we are looking at. So it was crucial for us to know these two. I think you can get the best information. We can write it here, but the best information is to know the flow, not what I will write here. I will use my English, yet you can use your own English. So I will not write that process here, but I can re-explain it again. Just get the flow of the information and then make your own writing, please. You said once a ripe pollen grain lands on a ripe and mature stigma, this is a pollen grain, and this is the stigma. This is the style of the stigma, and this is the ovary where the ovules are found. Once it is, the pollen grain has landed onto this stigma, it will absorb water and mineral salts from this stigma, and such that it germinates into a pollen tube. This pollen tube grows downwards. This is the style on the couple. As this pollen tube grows, the pollen tube is led by the pollen cell nucleus at the tip, as the generative cell nucleus is lying just behind the pollen tube nucleus. As this pollen tube grows downwards towards the ovary, the generative nucleus that is lying behind the pollen tube divides to form two meronuclei. These are the meronuclei formed. So this as the pollen grain, as the pollen tube enters the ovule, like I've been showing you here. Now the pollen tube is inside the micropyle of an ovule. Inside the pollen tube nucleus tip, the pollen tube will rupture. The tip of this pollen tube will rupture, will burst, such that the generative nucleus, the pollen tube nucleus disappears. Once the pollen tube nucleus disappears, it will give way to these two meronucleus to enter into this embryo sac. One of the meronuclei fuses with the egg cell to form a zygote, a diploid zygote, which develops into an embryo after fertilization. And then what was the second meronucleus that was formed fuses with the polar nuclei in the middle of the embryo sac to form what we call the triploid endosperm, which develops into a secondary endosperm that stores food for the seed after fertilization. So that is simply the information we must know about the process of fertilization in a flowering plant. We said what we are giving the source of information on which you can build on to write the better or the best information. This is Muto Online. Let's meet in the next video looking at the events that occur after fertilization and fruits. Thank you.